Good morning, everybody, and welcome to today's event. This event today is with FluoGuide. It's an introduction to the company. And with me today, I have the CEO, Morten Albrechtsen. Topic of today is a deep dive into FluoGuide's equity story. And then this morning, FluoGuide actually published their full year 2023 report. So Morten will take some highlights from the report as well. As always, the audience is more than welcome to post their questions during the session. And we already received a lot of good questions. And you post your questions just below the video window. And I will pass them on to Morten during the presentation. And we will record this event. And then afterwards, we will release it. And I hope Morten will release it on his homepage as well.
Yes.
So, but that's it. Morten, welcome, and please carry on.
Thank you very much for the introduction and for the opportunity to present FluoGuide and give you a highlight of what we do. So FluoGuide, what we do, we improve the outcome for patients with cancer, and we do that through precision surgery. First, our disclaimer, so please make your own assessments. So I would like to, in 3 slides, to really tell you what is the need here, how we solve it, and also what you can expect for us the next coming years. So first of all, you've seen that before. I'm sure that unfortunately, about 18 million patients every year get diagnosed with cancer. What is not so known is that about 80% of them will undergo surgery sometimes during their disease. And unfortunately, half of them will have recur of the cancer locally after surgery. It's not because the surgeon is poor in any way, it's just because it's really difficult, because today they use visualization, their eyes, and palpation the fingers to see if they can see more cancer. And there is no wonder that actually this 50% of the cases actually have and recur locally after surgery. So the solution to it, and that's where we come in, we call our lead product HC001, and it comes out of our UPAR targeted technology. It has 2 benefits or 2 effects actually. One is that it binds to cancer and light it up. So the surgeon during surgery can see it because the cancer lights up, and they can remove the cancer precisely. They are sure they get all cancer out, and they still can spare the normal tissue, but of course that does not light up. Whereas it makes it much easier for them to remove the cancer first time. The other aspect of it is what we call photothermal therapy. It's an property of the molecule. When you light on it, it will heat up, and when it heats up, it will actually destroy the cells bound to. And when it's very specifically bound to cancer cell, it will bind and destroy the cancer cells specifically when you light to it. It can be used in a couple of applications, and one is if it's difficult for the surgeon to remove all cancer, so hard to reach a cancer, that's a possibility that we can offer that for the surgeon. They can treat the patient as well for this cancer, and also it's a way of improving the surgery in the sense that they can kind of clean up the cavity after the surgeon has removed all the obviously visible cancer. And this is a very interesting dual effect we have in the molecule.
So, Morten, if I can interrupt you very shortly, because
please.
This is an introduction to the case. So just to be very specific about the UPAR receptor, this is very present in solid tumors, as I understand it. Could you elaborate a little on the UPAR?
Of course. I will come to that in a few slides, if I may.
Okay.
So I'll take it there. Then I go into the details of UPAR. Because if I first may just say what you can expect of the next, during 24. So one of the key things for us is that we want to advance the product, our HC001, to the market. That is going both for aggressive brain cancer and also going for head and neck cancer. It's important for us to do that as fast as we can, but also so predictable as we can. And one of the things with predictability is clearing with the regulatory agencies what is needed, the endpoints, what do they require for granting an approval. So this is work we are going on right now, and you will get more clarity on the design of the trials we want to do for securing our product get to the market. Then we enhance the benefit. We do that with photothermal therapy. This is an obvious way of doing it, but we do that through collaboration with equipment manufacturers, where we actually are able to get to cancer that is not reachable today, and by the technology we can actually help the surgeon to remove that cancer as well. Then we want to expand the number of patients. We do that first with head and neck, but also other brain cancers than high-grade glioma. and expand that into larger number of patients. And then, as Claus alluded to, on the uPARs actually go across all the solid cancers, and we could move into other indications as well. And then last but not least, we prepare for commercialization, and they have two arms. One is preparing the go-to-market with reimbursement, discussing with key opinion leaders, and building endpoints into trying to support reimbursement. But it's also to secure the optimal partnership, which we will have forthcoming. And I think we can actually do it in the sense we jump a little bit around, but we take the uPARs, as Claus alluded to here. So the uPAR is the receptor we bound to, and what is specific with uPAR is that it's an proteolytic enzyme, it's called, and enzyme system, and it means that it breaks down normal tissue. So when cancer want to spread into normal tissue, it would chew it up with uPAR, and then the cancer can spread into it. It's therefore often expressed on the forefront of the cancer, and is proportioned to the aggressivity of the cancer. And as you alluded to, Claus, I mean, it's very interesting with uPAR. It's both specific to cancer, but it's also expressed extensively on more than 8% of all solid cancer. Well, we started out with brain cancer. We could have started with a number of other cancers, but there's a lot of factors, and they was actually positive for brain cancer, and that was why we started out with this. If I can explain how we do it, bound to the cancer, we have a freeze-dried powder. It's reconstituted prior to use. Then it will be injected into the patient, and then it will circulate in the body, and it will bound to cancer. And then that will happen the day before surgery. Then at surgery, the surgeon can switch on the light, and they can see the cancer light up and help them during surgery. So that's very simplicity how we do it. It's a drug to start with, and that is under the regulation we are operating. And if I may be just a little bit technical here on FG1, FG1 is a molecule we are working with, and it consists of two parts. One is a uPAR targeting part, which is a peptide, and the other part is IgG, which is a fluorophore, and those two actually consist of FG001. One of the interesting things with it that it have the same spectral characterization as uPAR, as ECG, and that means that it fits to all equipment, so it's compatible to all type of equipment. And another aspect of it is that another aspect of it is that it can see deep into the cancer. So you can see it on the images below is a patient that both have the first generation product as well as ours, and you can see it much deeper into the tissue. So if I may have the video, me. So what you can see here is a surgery where you can see the cancer lights up in the middle, and you can see the cancer lights up in the middle, and you can see the brain around. And what is very clear that you can see the cancer there is from one of our early patients, and it was one of the very impressive images we saw, which is quite convincing, and it convinced really the neurosurgeon and also other surgeons we've been working with.
Morten, are you coming back and commenting on the very good data you received last year compared to the traditional treatment today? Or should I ask a question right now, because it's
just go ahead, Claus, and ask.
Yeah, it's okay. So because it's quite obvious if you compare it to the traditional therapy or the way you do it today, you showed it a small picture. It not only it's very easy for the surgeon to actually find the cancer cells, so you de-risk recurrent accidents for the patients. Isn't that true?
Yes, that's true. So we have very good data on 3 indications. So we have demonstrated in high-grade glioma, the aggressive brain cancer, and we have it in head and neck, and also in lung cancer. And we are moving into this step. So the first step is demonstrating our product is well tolerated. This we did in high-grade glioma, and then it goes for all the indications. The next step we then have to is demonstrate that it lights up cancer. We demonstrated that in all 3 indications, that it light up cancer extensively. And then the next step we do is we have a benefit endpoint where we show it actually, because one thing that lights up is not enough for getting it approved. We also need to show that it have a benefit to the patient, both to get reimbursement, but also getting approved. And this we have done in aggressive brain cancer, the first step, where we show that with the completion of resection, I mean, when they took out the cancer, was better when using our product. I'll come back to some data on this one. And then the next one is doing this in a larger scale, a phase 3 trial, where we can demonstrate that actually in a larger scale we can do that as well, and that's where we need to get the approval. So this is kind of the 4 steps we have to do, and for each indication we have to demonstrate a light up, have a benefit, and then prove it in a larger scale, and that we did in a classic brain cancer. This is one of the examples where we show with UPAR specificity. And one thing when you have a target, it's important that it mark the cancer and not the tissue around the cancer. And what you see on the left side is some tissue blocks with the cancer inside, and you can see that actually the cancer lights up in these tissue blocks, and it's prepared in 2 different ways. And this is very strong data in the sense that you really don't want to see that different part of the normal tissue lights up. You really want to see the cancer light up specifically, and that's what happened here. On the right side of the image here, you see it as a pathologist would see it. I mean, the pathologist is the one that have the kind of the truth for the surgeon, and they have the image in the middle is the one that they use as standard of truth. Then you have the UPAR expression that is correlated to the cancer, which we scientifically would expect. And then on the left side, you see our product FD001 also mark up the cancer. So really nice data that demonstrate that UPAR and our FD001 is actually specific to cancer and working as we expected. But of course, it's nice to see. Then there's this aspect of it also, the photothermal therapy, and that is when you light on it, it will heat up. And on the graph on the right side, you see different, and it's in mouse data, this one, and you see if you put in our product and you put in a laser and light on it, you will see you get a temperature. It's blue, dark blue curve at the top of them that is beyond the tissue damage. And this is important because if you have a cancer, then our product is bound to the cancer, and we light on it, it will heat up and it will destroy the cells. The other curve is below tissue damage, and that is if you have no product, or if you have no cancer, or you have just saline, I mean nothing, placebo. So it means basically that you could just light with a laser on the tissue, and then all cancer will be destroyed, but the normal tissue will be preserved. And that, of course, is really a great feature. Now we're moving that one into clinical testing to demonstrate how well that would work in humans.
In best cases, Morten, would this lead to surgeons lose their jobs?
Yeah, some surgeons ask for that, exactly that question, Klaus. That is a very good question. No, not really, because one of the things we quite concerned about, you can say, or attentive about, is that we don't want to compete with the surgeons. I mean, you will always, I mean, you have 3 treatment modalities today, basically surgery, drugs, and then some kind of radiation. And for most cancer, what you often see is that if one of them improve, I mean, you still use the 2 other modalities because cancer still recur and come so frequently back, or are not completely removed the first time, that you would like to use all treatment modality. So you often see that it's just added on top of the other ones. So really what we say, well, our mission is help the surgeon to make the surgeons better. And the key reason for that is that, as you saw on the very first slide, 80% of the patient gets surgery and it recur after surgery. And they're really an underserved group because there are not so many new products for the surgeons, and that's where we see a gap that they have a big need, and well, we can, and there's not a lot of them to help them with that need. So we really see it's an interesting thing to help the surgeons. And no, they will not be what can I say, unemployed, because they will really use the photothermal therapy together with the surgery, and we really get the surgeon to shine better, you can say, in that sense.
So even though you have a new technology, you would address the first line patient group. So first line in the sense that you use the common treatments like surgery, radiation, or chemotherapy, compared to some of the new technologies that is actually the group of others and normally address third line, a very patient. Yes. Yeah, you know what I mean.
Yeah, exactly. No, that's typically when you do a drug, then you get from the back, you can say, so to speak. Yeah. So you take the patient that have been treated with all the alternatives that have proven work, and then you start from the back, so to speak. No, we want to help the surgeon where the surgeon have the problem, and the surgeon have the problem with removing all cancer. So basically, we go in where the surgeon is in play. We help the surgeon. And you can say that's absolutely first line. And surgery is important when you have a localized cancer. Surgery is more important, and it's I mean, because really you want to do what you want to do with the surgery is that you want to remove all cancer the first time and take it out. So when it's first is spread, surgery become less important, and you more use drugs because they go to the entire body. So surgery is really for the localized cancer, but we help the surgeon wherever they operate, and also we want to help them on cancer they cannot treat today, because that could for their point of view, what can I say, be able to help more patients, and for the patient to have another treatment modality added to it. So this is a way where we can actually help more patients and more stakeholders, so to speak.
Yeah, especially the high-grade glioma. Well, you really don't have any treatment, isn't that right? It's really it's not a nice disease to have.
Well, I'm not saying you don't have a treatment, but you have treatment, but it doesn't work very well. So unfortunately, and I come to this here in the slide, that about half of the patients are deceased in 14 months. So unfortunately, it's a very severe disease, and despite the treatment you have, you can only keep half of them alive in 14 months. But if you have this, and this is because it's difficult, because the small image you see on this slide is the cavity of after the surgeon have removed the cancer. And here you should really, the surgeon should decide if they want to take out more tissue and be sure they have all cancer, but risk to disable the patient, or they should leave something behind with the risk that the cancer recur. And if you have the malignant cancer, I mean, then you're very concerned of leaving something behind, because they die unfortunately quickly, and less concerned on adverse events. I mean, you're also interested in this, of course, but less relevant. But the malignant cancer, they also have a curve. The cancer takes longer time, but they have a problem with the side effect, and that's what you really would like to avoid with them. So the solution for both the malignant and non-malignant cancer, that is increased precision, basically, and that's what we offer. So our tool help both the malignant and non-malignant cancer, two different ways though, but with the same objective, namely getting more precise surgery. And if you look at the numbers, and I think this is important, is that you have the malignant cancer, you have total of primary cancer that derive from the brain. You have about 170,000 new cases in every year in US and Europe, and 15% of them have high-grade glioma. So it's only a fraction of the primary brain cancers. The double number have other malignant cancer, and then you have quite a lot, 3 times as many that have non-malignant cancer, for instance, meningioma, which is half of them, and I come back to meningioma. So the reason why we start with high-grade glioma is just to get into the brain cancer. It's not because we want to stop there. We want to treat all the brain cancers and help them all, but do it in steps. Then there's another group that is frequently left out, and that actually cancer come from other locations. That could be lung or breast or other places that spread up to the brain, and they have the same problem as malignant glioma, namely that they're quite aggressive and that the surgeon want to remove them. And then we can also help actually for a large portion of them, and they are in numbers quite a high number, and this is number for US and Europe. So the whole market for brain cancer is quite a large market, and we just go in high-grade glioma, which is a very tiny little piece of it to start with. And coming into data, I mean, we have very good data on this is a brain cancer, all the brain cancer trial summarized in one slide. So we had 2 trials that has been conducted. They all been looking into high-grade glioma as that expected high-grade glioma that should undergo surgery. The first part of the trial was a safety trial combined with dose finding and the proof of concept, where we look at if it light up cancer. The second part of it, that was with 40 patients. The second part was with 24 patients, where we looked into randomized between the 5-ALA and our product. And the 5-ALA is this product, first generation product, that is based on visible light. And the issue with visible light is that you can only see it 1 to 2 millimeters into the tissue, compared to near-infrared light, you can see 1 to 2 centimeters into the tissue. And you can clearly see this for the images here, where on the left side you have a 5-ALA. The patient has been treated with 2 products, and when you switch on the blue light, you can only see the 5-ALA, and when you switch on the near-infrared light, you can only see FD001. So on the right side, you have the blue light. You can see that you cannot see the cancer, and on the left side, you can see where the cancer is within the ear, because it's located deeper into the tissue. And in this data, we have a completeness of surgery. So all patients that were treated first with white light, and then it was checked with the cavity, and we could see that all patients had additional cancer to be removed with the 001. We saw that FD001 was well tolerated, very well tolerated, in line with what we saw for other studies. and then we could see very deep into the cancer. So really a very good starting point for doing the regulatory trials, which we are now planning, and then was where we were looking into as well to combine potentially with the photothermal therapy, so we could add to the claim. This is why we have what can I say, done adding in the photothermal therapy, and that was basically because we got a grant last year that enabled us to do that in the spring last year. So as you may recall from before, I mean, the meningioma was a non-malignant cancer, but it actually constituted quite a high number of the cancers in the brain. And it was interesting because a 40 patient was suspected to have high-grade glioma, and one of them had did not have high-grade glioma, but a meningioma. So in this case, we also treated the patient both with 5-ALA and FD001. And as you can see for the top line, it's really hard to see the cancer where you can see it on the green in the right side with FD001. And if you look at the bottom row, you will see it on the back table. You can see there's some shine from FD001 treated cancer. There's nothing from normal and blue light. So it's really quite nice because it looks to that FD001 works across a different brain cancer indication in comparison to 5-ALA that only work on a high-grade glioma and where you cannot see so much into the depth. So really a quite nice position we are in, we think. And if I just summarize the plan for the brain cancer, we confirm the regulatory trials, we do assessment of the photothermal therapy, expand the label into other indication, and then we support for reimbursement. That is what you can expect us this year. Then I have the head and neck, just briefly. Head and neck is a little bit different than brain, but they also share different aspects. So you have the top part of the head and neck cancer, then you can see, and then you have the more bottom part of it, see it down into the neck. And those into the neck, the issue there is you cannot reach them by the equipment today, so you they're not eligible for surgery. So what you would do is that you would put the patient forward to chemoradiotherapy, where you actually both give them chemotherapy and radiotherapy. And the big issue with that is that the patient, when they get radiotherapy, is that they have to pull out the teeth because otherwise they get disease in them. So the patient really don't want to have chemotherapy. And you also have the same, if you remember from before, when you show the markings, I mean, when you see the tissue and that's cancer around close to the marking, then they have to undergo chemoradiotherapy if you have that finding in the study or in the surgery. So really what we help them to, again, is more precise surgery, reduce the number of close markings, and access patient that is not accessible today, and that's accessible because we can give them more precise surgery. So really we can help them all. And there's almost a similar number as brain cancer, quite a large market, unfortunately, for head and neck, and it's much larger in the third world. Here is only on the left side, only European and US, and then on the right side, you have the world, but quite a high number of patients, unfortunately. And if you look at the data, we have the first step here, we demonstrated it work on 16 patients, so we're very well off. And if you would show the video, then I can just show how it look like. So here what you see, now the surgeon look for metastasis in the neck, and they can see this is where they can see there's one metastasis there. Of course, that's a great help for them. They can see this kind of colored the image. So that's where they can find the metastasis. Here in the image, you see it on the right side as well, how they look at it. So yes, so I think that is very much it. I think
it's quite obvious.
Yeah, it is, and it's quite helpful because if you look at the tissue around, it's really difficult to see where it should be otherwise. So this is one of the aspects of it. But one of the things we also saw in the head and neck in the study, where there's several indications, where this with a marker that was finding the metastasis, but also get access to patients that they could not be accessible. So there's a lot of different aspects of it, and that is actually what is quite interesting with the head and neck. That's a multiple application. Another aspect of it, which is quite interesting, is there's multiple partnering opportunity because more or less all type of surgical equipment is used in the head and neck one way or the other. Microscope, endoscope, robotic surgery, but also the back table histology equipment, and that includes more or less all, or it does include all the large medtech companies in this pool. So summing up what we do in the head and neck, I mean, we initiated the phase 2 trial with the benefit endpoint. And what we mean with this is that it's an endpoint that potentially is supporting for regulatory, so to be trial positive, and we are looking into the multiple applications. We have expand and prepare for partnership and as well for reimbursement. So more or less the same thing as in the brain, but with a tweak on. But moving it forward to the market is a key thing. And that is summarizing really FluoGuide here in a nutshell. I mean, we have had a very good development until 23. We demonstrated that our product was safe and well tolerated with proof of concept light up in head and neck, brain and lung cancer. We have a clarified endpoint we need for regulatory support in brain, orphan drug designation in US for FD001 for aggressive brain cancer. We had the photothermal therapy grant, and we had the IPO in Spotlight in 19, and then moved to First North. And then what you will see happening this year, next year, is that we will have the design of the registration trial settle in this year, and we have it initiated next year for an imaging agent in high-grade glioma. We have the head and neck trial initiated in the result next year, and as well we'll have the photothermal therapy and the first clinical result next year. We would have partnering deals done this year, and as well further next year, and as well the marketing preparation. So really we are quite good shape, building on a strong platform we're generating since our IPO, and now we have kind of next phase, you can say, which is much more commercial and regulatory in aspects, and this is what we execute on now.
Thanks a lot, Morten. There's a lot of good questions, and especially we have one person in here who have a lot of questions to you, Morten. So I will try to read some of them up. Some of them you already answered, but just to take them for the audience in general. So if we start by the HGG and your photothermal treatment, he asked here, has the correct laser been found for PTT? And is this laser good to go, or does it need to be clinical approved? And how is PTT preparation work going on? So,
yeah, no, it's very relevant and good questions. I mean, I think we have found the right laser, and it would be good to go, and we would confirm that in first half, which laser it is, but we are making some tests at the moment to confirm that is the right one. But if it show out like we do, we have a really nice one that actually is all the regulatory work is done on it. It's so ready to really plug and play. And then more importantly, also in the partnering of the photothermal therapy will be slightly different than the partnering in the imaging agent. In the imaging agent, we want to partner with big medtech companies having surgical equipment that can see our product and then have multiple partnerships. In the photothermal therapy, we want to partner with one laser company exclusively, and then we want to have that together with our product being delivered as OEM systems into the big medtech companies. So it's a slight two-step partnering, you can say. And for the first part of it, identifying laser and build this product with them would be secured shortly. So in first half, you will hear more.
Good. And as I understand, you're of course looking into partnerships. And you have different models there, Morten. Maybe it's too early to tell something about what you're going for. But as I understood you're looking for a global partner on the laser part, but imaging partner you can find different partners.
Yeah, I mean, no, on the laser part, we want to have one partnership with one laser supplier, and then we want to together with them do an OEM. It's a partnership with multiple of the large medtech companies, so we can put that one into a microscope, into endoscope, into whatever they have. But it's a kind of a module we can put into their systems.
Good.
In the imaging systems partnership and surgical equipment partnership, we are looking for global companies, and then we're looking for companies where we have, for instance, in the head and neck. What I was trying to explain is that we have multiple applications, and that enable us to make multiple partnerships where we can have a partnership with one type of equipment manufacturers in one application, another one in another application, or even combine it. So it actually open up for multiple partnership. And what we have done since the very beginning is that, I mean, the partnership is as a dilution of our assets. Often we have to sell off some rights or share the profit upside with a partner. So we can either get dilution by financing, or we can get dilution by partnering. And what we have been looking at since the very beginning is how could we maximize the value generation without the dilution, with the different partnership models. And we've been talking with partners from the very beginning, because it's very important for us to understand what they want and what can be done. And what we see over the last year's time is that they're much more forward-leaning into do partnership with company of our type, because it's they know what they want now, and also they what can I say, they can see that it's a time for them to move to now, and that means that they're much more in partnership mode than they were for just a few years ago. So it's a very interesting development we see. And in the terms of the imaging agent partnership, we want to have multiple non-exclusive deals, and it will be very much a medtech-like deal, not like a drug where we get we will expect to have a big check written out around launch, not around partnership when we sign the deal. So it's not like a drug where you typically will sign on a lot of upfront payment now, and then you will kind of more or less sell the company. We will not. We will co-develop it with a lot of these partners down the road, and we will add more benefit and more applications to it together with the partners. So really co-development with them, you can say, and then in the end they will sell it in the indication. So it will be much more long-term marriage, you would say, than a kind of sell-off of assets.
Interesting, to maximize the value. That sounds really interesting, Morten. So this question here, and you probably changed that in your recent update from January, but this question here from one in the audience. So you used to inform the investor community about PTT and FG001 for different indications. Will as treating patients, will you do that? Will you still do that, or do you want to inform the market different in the future now that you changed your strategy a little?
Mm-hmm. Yeah, I mean, generally we have a principle that we want to inform the market as much as absolutely possible. One of the reasons for us that we did inform the market so frequent under the first trial was because when the hospital they do the trial, they know the product is used and not in the patient, and they know if it lights up or not. And we cannot kind of hide it, because you have a whole hospital system that walks around with nurses and doctors, and so when they come in from other types of doctor come in and see the surgery, and suddenly the whole hospital will know what's going on. And for simple, because we want to, of course, as being publicly listed, we have to secure everybody have the same knowledge. So we was very forefront leading in informing the market about the result of our trials, because then all have the same information. And it looks, when we look into the future, it will most likely be similar kind of studies that will be done, where we will kind of be more or less forced to have a frequent information of the market due to this insider situation. But there would be some of the design for the phototherapy application that where we will not know the result until the end of the trial, and therefore we cannot inform the market about what will go on. So it really depends on the design, but generally speaking, for the early trials, we will be very frequently information to the market, or deliver very frequent information to the market.
So if we take a little about the market sizes, Morten, and if we take your statements in the latest strategic update with PTT and for glioma, brain cancer, what is the potential market size? Because I know it's a rare form of cancer, but on the other hand, you could probably take a big market share.
Yeah, I mean, I think I would like to allude to the numbers a little, and there were all the numbers on the slide I presented, so you can see them there for reference. But in general speaking, we have about 300,000 patients with the brain cancer every year, and about the same with head and neck cancers. And if you look at the price, that is currently kind of the price in image agent, I mean guiding surgery, that's about $5,000 approximately per treatment. If you look at the photothermal therapy, it goes up to about $50,000 per treatment, or even up to $100,000 per treatment. So you would see that adding a photothermal therapy claim to the label will increase the price target, or the benefit and the price target, significantly. And that's of course why it was interesting for us to see if we can get that claim included somehow into our labeling. But if you just take, and I say just take the image agent price under 5,000 and multiply it with the numbers, it's a quite interesting market already. And if you then add lung, colorectal, breast, and so on, that's a huge market potential. And we have to be we have to balance. We have to go in and do something successfully and then spread cleverly, rather than to go for everything and then do nothing. So that is the balance we have to take.
And it's quite unique to use the UPAR receptor, as I understand it. Not a lot of companies do research within this field, isn't it true?
Yeah, that's true. That's true. I mean, it was actually invented back in time. Daniel, I think he was called. That was kind of one of the fathers, you can say, of UPAR back in time. Unfortunately, he's died now. So it is a science that is kind of evolved from this part of the world. But I think there's a number of companies that actually work with it on different processes. What I say, like a QSide on radiation and PET tracer, and you have as well, you have drug development companies also doing it, and you have drug companies in the past. So there are a number of companies working with the UPAR and try to do treatments out of them, but no one in the surgery area.
No, that's quite interesting. There's a question coming in here. Considering the medical principle, eliminate cancer tissue and preserve function, did you find any difference in clinical outcome as compared to the classical neurosurgical approach applying intraoperative or neurophysical monitoring? Sorry if I didn't pronounce it the right way, Morten, but you probably understand the question.
Yeah, I mean, that's a very good question. I mean, yes. Well, brain cancer is a difficult one, because if you in a brain, if you have a high-grade glioma and you take an additional tissue out of the patient with, let's say, 1 centimeter around the cancer, you will demonstrate that the patient will have a better surgery. And you would demonstrate that they will actually, the time for them to have recur of the cancer will be longer, so you can prolong it. But the price you pay is that you have increased disability, because and you have a decreased number of side effects. And then what it means for the patients, actually, in the end of the day, they will have a poor treatment after surgery than they would have had if they have had less good surgical treatment, because they're offered less chemotherapy and less radiotherapy. So the overall treatment actually worsened by having this additional cancer removed or tissue removed. That's one aspect. The other aspect of it is so just removing as much as possible is not the objective. The objective is removing the right thing. Then coming to the measurement, that is, I think, the question alludes to. One of the things is that the surgery itself actually blurs some of the side effects you get after, because when you do surgery, you often get seizures. When you get kind of epileptic, like you can get other kind of brain disability. You can lose your sight. You can do other things due to the surgery. And then disappear over time. So separating what is the cancer, what is the surgery, and what is the drug is difficult. You need to follow up the patient for a longer time. And this patient has been followed up for 24 hours after the surgery only. So we don't have those data for this patient. And then the question would be, should we collect those data for the patient, or should we rather look at the photothermal therapy and collect those data for that? And that's what really we tend to prefer, that the photothermal therapy will have a clearly treatment benefit in the patient, and it will be much more, in our view, sensible to actually collect the data and spend longer time doing the trials with photothermal therapy than fluorescence-guided surgery. But that's just how we think at the moment. So it's a difficult one to prove. That's my simple answer.
Fair enough, Morten. There's another product on the market called Gleolan. How does your product differ from Gleolan?
Yeah, Gleolan was this called 5-ALA in the slide. Okay. So that was the one we compared up against. And the way it differs is Gleolan or 5-ALA is a first-generation product based on visible light. So it had two kind of properties. One thing is that it's used only visible light, and that means you cannot see it very deep into the tissue, only 1 to 2 millimeters, compared to near-infrared light, which we use to make our product visible. And there you can see it 1 to 2 centimeters into the tissue. That's one aspect. The other aspect is that the molecule that is present in 5-ALA is a metabolite of the product. So it means that the cells generate this metabolite that's kind of form a cloud around the cancer. So it's not as specific to cancer as binding it to the UPAR. So there's a number of things that is different from this product.
Thanks a lot, Morten. And if you have any further questions, please post your questions and I will pass them on to Morten. But there's a question coming in here about financing, Morten, with those plans you have right now. And you mentioned that in your presentation, but you can probably repeat it. Are you fully financed for 2024 with those plans you have today?
Yeah, we would. Well, I mean, what we with the plans we have now, we can move ahead until, let's say, about Q4 or something like that. Then we would need to have some financing there. So we have no kind of urgency in financing at the moment at all. So we have time to wait and see for the market. We have time to see some partnering coming in and kind of establish some of the things in peace. But of course, as I always say, we always sleep with our boots on. We're always ready to move if something change, opportunity come up, and we can do it in a good way for our shareholder. We will, of course, do that.
Morten, do you have any opinion on, is it easy to find money for the moment, or is it more, is it hard to find money for the moment if you go out? And if you have a good project, you can always finance it, but what is your opinion on the financing market?
Well, you come into my favorite topic right now, one of them. I mean, yeah, I think as you said, if you have a good product, you can always finance it. I mean, one of the things we've seen for many companies, unfortunately, here when the interest has gone up, the capital become more expensive. And what has been important for us is that we've been very lean from the very beginning, and that has been an explicit decision because I mean, when we started, it was not. So what can I say? The cost of financing were not that high, but so we didn't have to be lean at that time. But actually, we prepared for the worst days, you can say, on the good days, and that was actually, what can I say, seen backward, very, very, very clever of us to do so. But I mean, it was done deliberately. It's not something we just done for purpose. It was done by purpose. So I think that we always want to keep lean until the stage where we are quite advanced, and there we of course can change around. We should change around at some point in time, but we don't need to do it so quickly. And that is a huge risk mitigation thing for FluoGuide, because the basic cash burn for us is very low. So we always have a possibility to not be squeezed in that sense. So that's one aspect of it. The other aspect of it that we have seen in particular in Sweden is that been some rights issue that's been heavily discounted and very, very destructive for the financing. So one of the things we also always keep in mind is that we have a long-term view of FluoGuide. We have 10 years horizon in our what can I say, time perspective, and we want to do funding that support our development 10 years ahead. So doing this very dramatic, destructive fundraising, we don't want to do them. We want to have always fundraising that we support, what can I say, long-term objective for the company. So yes, we don't want to go there. And we've shown that all the time that we haven't gone there, and we don't want to go there.
Thanks a lot, Morten. And I don't think there's further questions coming in. So by that said, Morten, I want to say thank you to you for a very thoroughly and into-depth presentation of your company, FluoGuide. And I know we're going to see you soon. And for the audience, we will post the video when we finish the video this afternoon, and you will find it on FluoGuide's homepage as well. So by that said, I hope you will all enjoy your day and you enjoyed the presentation. So have a nice day.
Thanks for having me here.